So this is a rather informal introduction of one of my esteemed persons I've ever encountered in our community. There are public figures, and they're really important public figures. And Dr. Mike West, who's going to be speaking now, is a really important public figure. How I encountered Mike West, he was the first stem cell scientist I had ever spoken to when I was involved with a very complex and unusual court case related to human cloning about 20 years ago, which I'll spare you that well-worn story. But Mike was my introduction to a field called somatic cell nuclear transfer, therapeutic cloning. He is an absolute pioneer in the field of regenerative medicine and stem cells. He was a subject matter profiled in a book called Merchants of Immortality, and has had an amazing entrepreneurial career at Geron, at Lineage, at Biotime, Advanced Cell Therapies. At each stage of his career, he's advanced the field. And his book, Immortal Cells, I have a special little bookshelf, either with special books or autographed books. And Mike's book, Immortal Cells, when he first saw the embryonic cells at the lab of Jamie Thompson, he said it was like nuclear fission in a dish. It was one of the key moments in his life that he saw cells that were basically immortal and could produce and produce and produce. And that stuck in my mind, and I realized if we can harness that, we can cure the world. How amazing is someone like Mike West that had the vision. So I'm so honored that he made the trip all the way from the West Coast to speak at our meeting today, not only to this live audience, but to the audience that are going to be seeing this on YouTube later and the much larger audience. So Mike, please come up and let's welcome Dr. Michael West.
Thank you, Bernie. Well, thank you very much. It's a pleasure to be here. Thanks to Bernie, Aubrey de Grey, and others, all of you that organized the conference. Already had a lot of interesting conversations. A lot comes of these meetings, and those joining us online, welcome. I'll be giving some information that comes from a company called Ajax Therapeutics. It's a public company, and so I need to remind all of you that I'll be making certain forward-looking statements that have risks and uncertainties, so I refer you to our filings with the SEC for more detailed information about the company. All right, so I feel the need to mention the demographics of aging just very briefly in one important context. Yes, we're all in this curve, and yes, the baby boomers are growing old. But the important point I wanted to make is that, as you've already heard, aging is really characterized by a problem of chronic degenerative disease. Chronic disease that impacts a patient for years and years is estimated to account for some 80% of all healthcare costs in the United States, and of course now is rapidly rising. And the economic challenge that poses for the United States, Japan, and many other countries around the world it's a tsunami. It's a disaster in the making. It's been estimated that the net present value of amounts owed to the aging population in the US for entitlement programs, most of which going to healthcare, is $200 trillion. It's a lot of money. The technology I'm going to discuss today represents the work of a lot of people. I'm going to talk specifically the work being done in our company. And it's targeted to directly addressing the problem of chronic degenerative disease and aging. And as I'll mention, maybe an unexpected benefit from this research is it provides, I believe, a really important insight into cancer. And so let me try to defend those outrageous claims. Well, how could something play an important role in cancer and in aging? Well, there's this old idea of antagonistic pleiotropy. Some of you may have heard of it. Bottom line is a theory proposed many years ago is that aging evolved in metazoans as it's called antagonistic pleiotropy. The concept is that genes or alleles or the expression of genes, the pattern of gene expression was selected for optimum survival early in life, but those same changes or alleles or genes expression kind of a detrimental effect later in life. And evolution was really selecting for fitness of reproduction, regardless of the consequence later in life. Now, a classic example, which I spent a lot of my life working on, was relating to telomere biology and telomerase. So what we see is that very early after the egg is fertilized, cells start to branch out away from this immortal germline, which I'm showing you here in red, the lineage of cells that perpetuates the species forever. Somatic cell lineages, or body cell lineages, very quickly, within a couple of weeks, I think, for the most part, repress telomerase expression throughout the soma. And so a clock starts ticking. You may have heard of the telomere clock. So every time a cell replicates without telomerase, telomeres start shortening, like a burning fuse, leading to cellular aging and senescence. As an example of how this is antagonistic pleiotropy at work, here in this diagram, it's a bit busy, but on the left you can see the expression of telomerase, what we call the catalytic component of telomerase, TERT, telomerase reverse transcriptase. You can see the expression in ES and iPS cells. These are immortal pluripotent cells that Bernie just mentioned. And then I'm showing you here the lack of telomerase very early thereafter. What it shows here is diverse EPs, refers to embryonic progenitors that are branching out of the first cells of life, pluripotent cells. You can see they've already lost immortality. And then I'm showing you diverse other kinds of adult cells of all kinds, heart muscle, liver, and everything else, epithelial cells. But look at the expression reemerging in cancer. So the thought of antagonistic pleiotropy is, in the case of telomerase, is the benefit of turning off telomerase early in life helped reduce the risk of cancer, because cancer requires a lot of cell proliferation to evolve. But the downside, of course, is cellular aging. But cancers need to turn telomerase back on. And this is interesting. You've heard that there's lots of types of cancer. There's no cure for cancer because there's kidney cancer and there's liver cancer. Look, there are pan-cancer phenomena at work. Telomerase is one. Some of you may point out, well, there's some cancer cells you're showing me, Mike, here that have no telomerase expression on your graph. Yeah, but those are using an alternative mechanism to maintain telomeres called ALT. And essentially, all cancers except a couple have escaped this restriction, this somatic restriction of telomerase expression. Okay, that's telomeres. What's that got to do with the regeneration? Well, so back in, Bernie mentioned the early work that was done some 20 years ago on reprogramming by nuclear transfer. It eventually became to be recognized that there are genes that are the tools that the egg cell uses to reprogram a fibroblast back to the beginning of life. And the common ones, some of the common ones I've listed here, SOX2, Lin28, MYC, OCT4, NANOG, there's others. You can pick and choose depending on the cell type. But we did a study back 2010 showing that you could when you reprogram cells back to pluripotency, even aged cells, you can restore telomere length back to the beginning of life and reset the telomere clock. Okay. Now I want to bridge to this idea of partial reprogramming. As Dr. Eber-Katz just presented, there's a lot of interest in the scientific community in regeneration. And there are some animals that are developmentally arrested early in life maintaining a regenerative state that's really profound. And one example I'm showing you here in this picture is the Mexican salamander, the axolotls. For instance, you can amputate an arm or a wrist or a finger or whatever, and it nearly flawlessly grows back because these animals have developmentally arrested. Their clock of development is arrested very early in development. So how does that repression of regeneration work? As someone once said, could we make a salamander man? Could we make humans do this? If the mechanisms of regeneration are in existence early in life and are turned off, can we restore what we once had? Humans, early in our development, we have a profound regenerative potential in our bodies. Could we find a way of unlocking it? And so you can see where I'm headed. The idea then bridged to, could we use these reprogramming factors to take cells back to a regenerative state? So this has been called partial reprogramming by some. I prefer the term induced tissue regeneration, or ITR, because I think to make a real product out of this, we need to find a way of precisely understanding and controlling this reprogramming to induce regeneration, but not to take cells back to pluripotency. And so I'm going to describe for you, just in a few minutes, a very simple concept of developmentally regulated ITR, a precise means of regulating reprogramming to take cells in the human body back in developmental time to this regenerative state. I want to tell you about how we've been thinking we could do that. What would that state look like? Well, let's start with skin. In the case of multiple animals, rats, mice, humans even, marsupials, where the animals enter the pouch, still in an embryonic state where you can see regeneration. Scientists have carefully mapped when scarless regeneration gets turned off. And in the rat, you can see in this paper I've highlighted, it's around embryonic day 18. And so here you can see different animals comparatively where in their developmental time this is occurring. In humans, it's around 60 days, 2 months of developmental time. That's the embryonic fetal transition, EFT. What's significant about it is if you think 2 seconds about it, it makes perfect sense. That was a point in development where anatomists recognized that organogenesis is essentially complete. The human is, you built the human body in a miniature form. If nature turned off regeneration at about that time point, it makes sense if it's antagonistic pleiotropy. The human body is basically formed, it needs to grow, but organogenesis is complete. You don't really need to repeat the process. If it helped prevent cancer, let's turn that ability off, okay? Kind of makes sense. So I'm bridging here from the animal data to human data, the embryonic fetal transition. Well, we did, and I don't have time to explain how we did this, but we did a very extensive, like 8-year research project within AJAX to really understand this transition. In brief, what we did is we took hundreds of diverse types of pre-EFT cells that were still in this regenerative mode, human, of diverse kinds, and compared them with adult counterparts and fetal counterparts, and then did transcriptome, metabolome, epigenetics, ChIP-seq, all kinds of analyses to try to really understand this transition, the molecular biology behind it, and interestingly, it's consistent with the 2 previous speakers in that regard, a role for lamins, A and B, in this process, and the transition from the glycolysis to oxidative phosphorylation as well. Now here's an example of the transition. You've heard, as mentioned, you heard of the Warburg effect. Early in development, our bodies rely on glucose, glycolysis, and then we shift to oxidative phosphorylation. Cancers tend to revert back, the Warburg effect, back to the embryonic mode. Well, here's an example of a gene we think is important in this. It's called COX7A1, and on the far left you can see the expression of this gene in pluripotent cells. It looks like there's a little bit of expression there, but that's background microradiator. And then at 8 weeks, just as we're entering the transition to fetal development, this is actually all skin cells taken from the upper arm of fetuses and adults of different ages. You can see the expression of this gene turn on and then maximize as you reach adulthood. This is just a marker. Well, it's probably more than a marker, right? So how could we precisely regulate reprogramming of cells back to a regenerative state just before the embryonic fetal transition? Well, I just showed you a gene whose promoter is relatively well characterized. And so we're developing this as we call developmentally regulated ITR, which is more sophisticated, we think, version of partial reprogramming, where the promoter of COX7A1 drives reprogramming factors, in this case, KLF4, OCT4, and LIN28, which we have determined to be optimum for inducing a regenerative state in skin. So I think you can see how this works. These vectors would instill a reprogramming pattern of a gene expression until COX7A1 is turned off, and then the reprogramming factors are turned off. As an example, in addition to what has been described previously of regenerating mouse, this is the African spiny mouse, Acamese, and here you can see the expression of one of these genes, LIN28, B in Acamese versus normal mice. As an example, this is a really profoundly regenerating mouse, right? Amazing. Another way that you can more precisely regulate the induction of regeneration is just what we call segmental reprogramming using defined genes. And here is a hair regeneration study done with just the knockdown of COX7A1 and the introduction of LIN28, and then the combination of the two. And you can see the profound effect on hair regeneration in these animals. Now, this is a timeline. This is from Malova et al.'s paper in Aging Cell on the timeline of reprogramming, partial reprogramming. And I put into this timeline the expression of COX7A1. You can see that its expression drops to embryonic levels within a week of the, being exposed to reprogramming factors. The pluripotency is not seen for about 3 weeks, 2 to 3 weeks. And you can see here labeled TERT, telomerase, is expressed as cells re-enter pluripotency. So to get the optimum effect in aging, we would like to have telomerase re-expressed and reprogram cells to a regenerative state. Simple animals that have immortal cells that have telomerase and profound regenerative potential don't show aging. There is no Gompertz curve in many of these animals. And so we would like to design a technology that introduced telomerase and regeneration, but that, based on the studies we've done and what I'm highlighting here, we believe will require a separate addition of telomerase. I don't believe you can partially reprogram cells and tissues and induce a regenerative state and get telomerase unless you reverted the cells back to pluripotency, which has significant risks in my mind. Now I want to shift really quickly to the flip side of the coin. You remember I told you about antagonistic pleiotropy, right? Probably aging is a tumor suppressive mechanism. And I give you the example of telomerase. Telomerase is turned off in the soma, and reappears in virtually all cancers as evidence that this is a really important, the repression of telomerase probably had a lot of selective value in the history of life as a means of allowing us to live as long as we do without free of cancer, humans. Mice typically die in a couple years of cancer, right? All right, what about regeneration? What we've discovered is that like telomerase, upwards of 90% or more of cancer cells have reverted to a pre-EFT state. Oh my gosh, that wasn't known before. When it was recognized that telomerase is an important marker of cancer, became a hallmark of cancer. It's one of those segments in the wheel, you know, along with oncogenes. There's been this sense that cancer is kind of embryonic, the Warburg effect, right? I just mentioned. Oncofetal antigens are sometimes used in diagnostics. Here's just an example. Here is Lin-28 again in pluripotent cells. You can see some of these pre-EFT embryonic progenitors, EPs there, have a little bit of Lin-28. It's essentially often all adult cells that we could ever see, and reappearing in a lot of cancers, not all, but many. I think you can see, yeah, a lot of cancer cells have reverted back to pre-EFT state. Here's a couple other genes. I want to introduce you to the CPT1B. This is carnitine palmitoyltransferase 1B. It's a good marker of cells that have reverted back to glycolysis. And you can see on the graph on the right that pluripotent stem cells express this gene, as do these pre-EFT embryonic progenitors that are in the regenerative mode. But fetal cells, FC labeled there, adult non-epithelial as well as adult epithelial cells express very low or none of this gene, whereas cancers, sarcomas and carcinomas re-express it as an example of this Warburg effect. There's other interesting markers we found that are characteristic of the regenerative state. On this volcano plot here, on the left you can see all these PCDH genes. What are those? Those are the clustered protocadherins. So early on it was recognized that in early embryonic development and in regeneration, this goes back many years, there must be a way that cells can recognize each other. Cell adhesion must play a fundamental role in regeneration. And so what we've discovered is that these genes, which in the adult are only expressed in the brain for a different reason than regeneration, are expressed, not expressed in adult, are abundantly expressed in embryonic development and abundantly expressed again in cancer. So the way to think about these genes very simplistically is imagine if we, you know, Velcro. Imagine we had hundreds of thousands of different kinds of pairs of Velcro. So only, you know, Velcro 1, the two would stick together, and the Velcro 2, only those kinds would stick together. Then we could take a jet airplane, take it all apart, and put all those Velcros in the right place, put the pieces of the jet airplane in a big box and shake it, and out would fly a jet. Okay, overly simplified. You get the idea though. These genes appear to be playing a role in allowing the amputated limb of a salamander, for instance, to recognize the cells that belong together and reorganize themselves, even despite tremendous trauma. And if you look at those and a bunch of these other embryonic markers here in sarcomas and carcinomas, they're a hallmark of cancer. I mean, they're abundantly re-expressed in cancer. 100% of carcinomas that we've studied re-expressing these pre-EFT. Now, it's important to point out, I'm not talking about pluripotency markers. I'm talking about pre-EFT markers. If we looked at OCT4 or NANOG or any of these pluripotency genes, rarely they're in cancer. I think out of 1,000 cell lines we looked at, we found like 2 that had OCT4. All right, so this opens the door to some novel therapeutic and diagnostic strategies. I don't really have time to go into detail, but we're designing vectors that utilize the unique cell adhesion molecules re-expressed in cancer cells that are not in the adult from certain of the clustered protocadherin genes, and using a promoter that's embryonic specific like CPT1B to drive toxic gene products. So to target these vectors to cancer and then selectively destroy the cancer cells. And what's exciting about it is it's potentially a pan-cancer therapeutic strategy. Here I'm just showing some data. The antibodies alone to the embryonic clustered protocadherins can recognize cancer cells and statistically significantly kill them and not touch normal cells. Diagnostics. This was a study, not ours, and a published study looking for pan-cancer markers, gene expression markers. And I've highlighted in yellow here the genes that are underexpressed in cancers, multiple cancers. This is a meta-study, you know. Isn't it interesting that the ones that are underexpressed in cancer are adult-specific markers that we discovered as part of the CFT study? Wow. Here is an example of how you can see this in methylated DNA. The advantage of this modification of DNA, methylated DNA, is it's durable. So fragments of DNA from a tumor can go into the circulation, and you can do what's called liquid biopsy. You can actually find little snippets, little nucleosomes of DNA, and by PCR find them and detect them. And here you can see that the top 4 are embryonic pre-EFT cells. They're in this regenerative mode. And at the bottom you can see adult counterparts that are adult. And you can see they're undermethylated in adult. And but then at the bottom you can see these little tick marks. They're re-expressed again in prostate cancer, breast cancer, colon cancer, and so on. Pan-cancer. These are pan-cancer differentially methylated regions, DMRs. So we're really excited about this. Now, I'll make one last point. What I've told you so far probably won't come as a surprise. A lot of it's been hinted at and is where the field's going. I described how we think of regeneration in cancer as, you know, 2 sides of the same coin. I mentioned why we think these regeneration genes are turned off to help prevent cancer, just like telomerase being turned off. Re-expressing them could play a fundamental role in inducing regeneration, reprogramming, reversing the aged phenotype of cells. You get all that. This might come as a surprise, what I'm going to tell you next. It was a surprise to us. All right. There's a thing that people have been talking about for years called cancer stem cells. You guys have heard of this, right? I first heard of cancer stem cells back in the stem cell days when all this news was breaking about embryonic stem cells and so on. And I thought, well, everyone wants to get in the game, right? Now there's cancer stem cells. Well, as best I can piece together, is what happened was people recognized that when you have a breast cancer or a lung cancer or whatever, you use radio or chemotherapy to treat it, there are resistant cells that are not destroyed by the chemoradiotherapy. And this reminded, I think, some researchers of, well, you know, stem cells are relatively resistant to chemo and radiotherapies. The reason is they rarely divide. Bone marrow stem cells sit there in that niche in the bone marrow and maybe divide once a year. So and these agents kill dividing cells. That's how cancer cells are selectively destroyed. by alkylating agents and so on. So they thought, oh, these resistant cells are cancer stem cells. Then the idea was, maybe they express pluripotency markers. But then the field got confused because people started talking about what's called the epithelial mesenchymal transformation. On this diagram, you can see I'm showing you an epithelial carcinoma, these epithelial cells multiplying, making a small tumor and evolving. And then they do this transition where they become like a fibroblast. They start crawling around and metastasizing through the body. And when people were studying the epithelial mesenchymal transformation, people started saying, you know what? But those are the stem cells. And so the field's rather confused right now. But the thought was, and maybe still is, that the cancer stem cell, the mesenchymal cell, the one that's resistant to chemotherapy, is more primitive. Many people even say they express OCT4 and pluripotency markers. It's a more primitive cell, right? It's a stem cell. What I'm suggesting is it's absolutely backwards. So I don't have a time here today to show you all the data, but what we see is using these markers of the embryonic fetal transition is that the cancer stem cell, the resistant cell, is a cancer cell that has reverted back to an adult non-regenerative state. It's absolutely backwards. So the cancer stem cell has lamin A, an adult-like cell. The embryonic version of the cancer cells have lamin B1 and have a relative lack of lamin A. So it provides a whole new insight, I think, into potentially addressing this nasty problem we have of understanding these resistant cells and how to specifically target them. Because in the case of many cancers, we can debulk a lot of the tumor, but these resistant cells have been relatively resistant. So I scratched out cancer stem cell. They're adult-like cancer cells. Someone will come up with a more clever name than me, I'm sure. We at Ajax, I won't bore you with the clinical development, that we have 3 products in development, brown adipocytes made from pluripotent cells for the treatment of diabetes. Renolon is the application of reprogramming to induce scarless wound repair using developmentally regulated ITR, which I sort of introduced you to. EPRO is a cancer therapeutic strategy based on some of the things I told you. And then there's the cancer diagnostic aspects of this, which we plan to partner out. So in summary, I mean, we've seen some revolutions in biotech that had a lot of impact. Recombinant DNA started the industry. Monoclonal antibodies have been a platform allowing us to target many different antigens with a lot of clever strategies, a lot of immunotherapies. Regenerative medicine, since this conference started, Bernie, some years ago, has made significant strides. As you can see, some of the deals that have been done here. My previous company, Biotime, now Lineage, recently announced a nearly $700 million deal just for RPE cells for the treatment of ocular disease. This example of this is a platform because you can make hundreds of different cell types for many different therapeutic applications, and we'll see that in the future. I would call that the regenerative medicine 1.0 because 2.0 is now being born, and the ability to induce a regenerative state in a context of age-related degenerative disease, which, as I mentioned, is 80% of our healthcare costs, straining the economics of the United States, causing untold suffering to our population and our friends and our loved ones, is where the field is headed. And so with that, I'll just summarize and say that there's a lot of therapeutic and diagnostic strategies coming out of, we'll call it partial reprogramming research. The work that's done in the past by Dr. Heber Katz and others working on axolotls and planaria and all of this goes back now over 100 years. has led to improving understanding of how we can induce tissue regeneration. What's so exciting about this is, like telomerase and telomeres, a technology that originally started out as a way of treating aging has surprising and important applications in cancer as well. With that, I'll thank you very much.